Hello, everyone. Welcome to Socorro's webinar series, Coastal Observing in Your Community. My name is Abby Wakely, Socorro's Director of Communications, and I will be facilitating and moderating the webinar. Socorro is hosting this webinar series to discuss, highlight, and raise awareness about coastal ocean observing activities in the Southeast U.S. and beyond. Socorro is the regional coastal ocean observing system for North Carolina, South Carolina, Georgia, and Florida. We are one of 11 that make up the NOAA-led U.S. Integrated Ocean Observing System, also known as USIUS. Our mission is to sustain and promote observations that help keep you safe. This is a quick snapshot of the technology we support to accomplish our mission. All right, let's get to the reason you are all here, the webinar. During the webinar, we highly encourage you to ask questions for the presenters or technical questions for me by clicking the tablet look-in icon and typing in your question. I will pose them to the presenters after their presentation. We are recording this webinar today and will be posting the recording on the Socorro website and emailing it to everyone who has registered. We are very excited to welcome Scott Duncan and Veronica Ruiz Xomchuk today. They will be presenting long-term Lake Okeechobee harmful algal bloom and water quality monitoring with the Nav2 Sail and Solar Automated Surface Vessel. Scott Duncan has over 20 years experience with design and fabrication of custom automated surface vessels for ocean data collection. In 2000, he designed and built the first Navocean sail power prototype, the Mini Nav1, as proof of concept for the University of Washington Applied Physics Laboratory. He went on to develop numerous automated surface vessel prototypes of both sail-powered, electric propeller-driven, and hybrid designs. Scott founded Navocean in 2016 to pursue further development and application of the Nav2 Sail and Solar Automated Surface Vessels for ocean data collection. Veronica Ruiz Xomchuk is a senior research fellow at the Harbor Branch Oceanographic Institute at Florida Atlantic University. She received her PhD in oceanography from Texas A&M in 2020. As a postdoctoral research data scientist, Veronica has been responsible for the data flow and analysis for the harmful algal bloom assessment of Lake Okeechobee, the HALO project. This includes modeling, in situ monitoring, and remote sensing data. All right, Veronica, I am going to promote you to presenter. And so you should get a prompt about what screen you should share. All right, we are seeing the PowerPoint.
Okay, are you seeing it?
Yes, and I think the closed captions are on. If you just want to close them. There we go, perfect. So I'm going to go off camera and mute and we can hear and see you. Scott and Veronica, take it away.
Okay, yeah. Okay, thank you everybody for being here. I have been told that there is a very big attendance today, so we're all excited about that. I'm going to be starting a little bit, but then I will give the floor to Scott. So this is work that comes from a project that we got to develop last year over 2021, and it was funded by the Florida Department of Environmental Protection, and this is a part of it. And so Scott Duncan will be presenting the part about the Navocean vehicle. I will be talking more about data, the setup, and then the kind of like the brains behind this, Jordan Beckler, assistant professor at Harbor Branch. He's going to be here also for questions if there are more general topics and things. And so just going into it, here on the right side, you can see a picture, a satellite image of Lake Okeechobee. This is from last year, this is July, from Sentinel-2, and you can see a pretty widespread algal bloom developing in there. And you can see it clearly, and the resolution is quite high. But there are some buts with everything. And so some background on Lake Okeechobee. As you saw, the entire lake, it is pretty big. It is the 8th largest freshwater lake in the US. It is a pretty extensive area, and so to monitor it all, it's difficult. On top of that, it is shallow, and this means like this area and this shallowness means that there is sufficient fetch and shallowness to allow that frequent wind, wave, and current generated events resuspend the lake and make it very turbid. And so this also works towards eutrophication. The lake is eutrophic, but then that input from the sediment also affects that. And because of that, we have frequent algal blooms, and predominantly, and the major worry of this is microcystis, which is a cyanobacterium. It has a very particular color. But more than that, more than Lake Okeechobee itself, and the local problem, it's also affecting other regions because there are discharges from Lake Okeechobee that impact other places. And so as an example, I'm showing two publications that have come out from the last couple of years. The top one studies how the discharges from Lake Okeechobee are affecting Carenia brevis blooms in the west coast of Florida, and how discharges from Lake Okeechobee affect the development of blooms in the eastern coast of Florida. So this is something that is not only local, while the local community are very worried about that. And so also some background about that. The South Florida Water Management District has made a huge and incredible effort to increase monitoring in the lake. They have been monitoring several stations in this. Here you see a map of that, and they have made an effort to put even more stations. So in this map, where you see the yellow dots, those are the new stations that they have, and they have also implemented some kind of continuous monitoring stations in there. And so you see those are the asterisks in here. So they have a pretty good and nice coverage of the lake on what's going on. And from all these studies, and so this is taken from last year's report from them, they have identified that there is more concern of blooms happening in the north portion of the lake. And so this map shows the number of bloom events that happened, that occurred during that period. And you can see again, the yellow and reds are where we have concerns. And so in this context and in this problem, we got funding from the Florida Department of Environmental Protection to test some technologies and to try to better understand what's going on in the lake and how can we better predict the happening of this lake. So in this project, we really got to test new unique technologies that haven't been applied before. For instance, in situ kind of like photograph of cells to try to identify species with the autoholo, or the AZFP, which is a kind of an acoustic sensor for that. We've got to monitor sediments. Jordan Beckler is the one in charge of that. So we've had like a pretty neat data set for continuous sediment monitoring, coring, the deployment of a benthic lander. We look closely at the optical distribution of the water column. Again, Lake Okeechobee is pretty turbid, so there are a lot of complications with that. We even monitored air toxins and how those will affect people in the field. We used remote sensing products to try to establish better how harmful algal blooms are detected. We also put a couple of sensors in the surface and in the bottom of the L001 or North Tower. We are going to be talking a lot about that. And so some of the high resolution stationary time series that we have are from here. But today we're mostly going to be talking about the autonomous surface vehicle and how that vehicle helps us explain what's going on. And so with that, I will pass the floor to Scott. So he is going to be talking to you more about that, and then I will be back for the data. Scott.
Okay, thanks, Veronica. Yeah, so I'm going to be talking a little bit about the sailboat itself and the sensors that we integrated and what it was like to bring the sailboat to Lake Okeechobee and operate there for a year from the perspective of mission operations. And I just wanted to add thank you to the Florida Department of Environmental Protection and their Blue-Green Algae Innovative Technologies program, which is really where they helped us got the funding from to help us to do this. And their support made it possible to try this very innovative thing of never before tried on Lake Okeechobee, which was to bring an automated surface vehicle here for the first time. So thanks. And let's see, I'll go ahead and change the next slide, Veronica. Yeah, great. So a few things about the sailboat itself. The Nav2 sailboats were designed to be small and lightweight and non-hazardous by nature, so that in the event of any kind of a collision, the sailboat might lose, but we wouldn't cause damage or harm to anyone. And so that was one of the driving design characteristics, which meant we needed to keep the sailboat very small. You can see the small size of the design in the photo on the right, upper right. I included that one so you can see it next to 2 people there on land. And also that's out at the beach in Sarasota near Lido Beach on the west coast of Florida. And we did run some pilot project field tests related to red tide out there and fisheries as well. And that does relate back to the Lake Okeechobee work that we're doing. As the Lake Okeechobee harmful algal bloom plumes flow out through the Caloosahatchee Canal and interact with the Indian River, the lagoon, and the nearshore coastal areas where the Crennia brevis red tide interacts. But that's a sidetrack. So let's get back to the lake here. And you can see in the bottom right corner where the Harbor Branch speedboat there that was used for the research boat on Lake Okeechobee is a small boat, actually a pretty good sized one for Lake Okeechobee, but you can see how small the sailboat is by comparison. And if you look carefully, you can see a researcher leaning over the side of the research boat and collecting a water sample from just below the sailboat's hull where the sensors are located. And also, oftentimes get the question of, hey, are these boats, how do they do in a storm? What happens then? And they were designed originally for use in the open ocean and have been used in the Atlantic Ocean in the Gulf of Maine and off the Washington State coast in the Pacific Ocean. And the bottom left corner shows a photo that it's hard to get photos in the storms actually, but that one turned out okay. And that one was from, believe it or not, a mission on a short field test in Florida on the Banana River Lagoon, and that is near Cape Canaveral. And during that test, a strong storm system moved in and just happened to hit right onto that location. And so Vela got to ride out a couple of days of storm system. I think winds hit about 48 nautical miles per hour in that one. So that's the basics on the sailboat. Go ahead and move to the next slide, Veronica. Yeah, so considerations for bringing an autonomous surface vehicle to Lake Okeechobee. Definitely a new thing to do. And as we discussed before, one of them was to make sure that it was operating safely. And so that's one of the reasons for choosing the Nav2 boat in the first place. So we don't have to worry as much about things like object avoidance technologies, which are still really in their infancy for use on autonomous surface vehicles. And so we were able to operate outside of line of sight of observations and go fully autonomous for long range. And seaworthy, and that's notable on Lake Okeechobee because Lake Okeechobee is a big, broad lake. It's also very shallow, and when the winds pick up there, it's known for being a hazardous waterway with very steep waves and very aggravated sea states. So, and also capable of multi-sensor payload integration. And that was a big part of the innovative aspect of this program, was to see if we could really get a complex, comprehensive suite of sensors all integrated to such a small autonomous vehicle. So for community engagement, and I'll get back to that a little bit later on, a lot of local people really got involved and really appreciated the work that we were doing, which was great. But just to start with, we made sure to register with the Coast Guard and local notice to mariners to get their approval, and notified the Florida Fisheries Wildlife Law Enforcement. So they were looking after the boat for us as well. Although for the most part, when we did encounter them a couple of times, we had to remind them that it was out there, but that's okay. Thanks, guys. You guys did a great job looking after it. Greatly appreciate that. And Florida Atlantic University Harbor Branch put out a couple of press releases to local TV and newspaper outlets. And also one of the things is just, in the thought experiment of what are all the things that could go wrong with trying this, bringing this small sailboat to the lake for an entire year. And the three things that we came up with, which were our primary concern, one of them was lightning storms, because Okeechobee is right at the center of the state of Florida, and it tends to be a convergence zone for weather system from the Atlantic and also from the Gulf side, and a lot of vertical development there and a lot of lightning. Having now spent off and on over the course of the year some time there in Okeechobee, wow, it's impressive. But we did not have any problems with lightning strikes. Also, a vessel collision of some kind, or would somebody mess with the sailboat, and no problems there at all. And yeah, the local inhabitants kind of joking, but actually really not, because we had no real good long-term experience operating around alligators, just short field tests here and there for a few days. And what would happen over the course of a year, and for sure numerous alligator encounters during that time period. Lake Okeechobee, the center of the lake, the ecosystem's not doing so well, but around the perimeters of the lake, it does have a lot of tall grasses in the shallow flats, and that's where the ecosystem is still thriving, and there are lots of alligators there. And so it turns out that they probably came by and took a close look any number of times. Toothy critters tend to be quite curious. They also tend to be smarter than we think, and they took a pass at taking a chomp out of it. So that was good news. Next slide, please, Veronica. Thank you. Yeah. So the sensors that we integrated. So the Nav2 sailboats come with an AirMar for wind and meteorological measurements, important in the context of HAB developments on the lake. And we also integrated a conductivity and temperature sensor to get some basic physical water quality measurements. And a Nortec Signature 1000 ADCP was a fairly substantial item to integrate. And that's for getting the current profiles at different depths. And although it's a lake that doesn't have a lot of movement of water in it, but it does have slow moving currents, mostly generated by the wind. And the difference between surface and bottom and things like that can have a big impact on HABs development, we believe. So we integrated that. And also for HABs related water quality, a seabird backscatter to get the turbidity, which is so high on Lake Okeechobee. And so we understand how to that can affect the measurements from the fluorometer, the Turner Designs fluorometer that we integrated, which has the CHLA, CDOM, and phycocyanin measurements, which are highly useful for identifying algae concentrations in the lake. The CHLA gives general purpose algae concentration estimates, and the phycocyanin is useful for cyanobacterial type of algae, which Microcystis and some other toxic algae on the lake fit into that category. And then the O2 sensor from Andera, that ended up being very highly interesting because the O2 concentration goes up because algae, of course, is a plant. So when it grows, it sucks in carbon and it puts out oxygen, and that supersaturates the surface layer of the lake. And we could actually track when the algae blooms were actively growing fast. We took one measurement every 5 seconds, duty cycles for 5 seconds on and then 25 seconds off. And that created a good high resolution spatial temporal data set. And the Nav2 ASV is also outfitted with cellular and iridium communications, which was important for the lake because although there's cell phone connection over cell phone modem connection over much of the lake, actually a lot of it does not have not good enough for consistent relaying of transmit of messages. So the iridium was actually quite important to be able to transmit by satellite. And so we would send every 15 minutes by satellite a sample data set that showed where the boat was and a sample of the data measurements at that location and time. So yeah, Veronica, next slide, please. Yeah, so the sensors integrated, we took a couple of validation steps to make sure they're operating correctly throughout the year. One of them, you can see here, one of the observing locations, we'll get back to this in a moment, but the North Tower you can see in the left image there. And we would send the sailboat there because that location has a number of other sensor systems that were installed by the HAB assessment of Lake Okeechobee. Our HALO overall project had many other aspects to it, and many of them were located right there. So we could get the validation samples coincident with the location of other sensors. And those lab samples were, the water samples were validated back at the lab. Next slide. Great, and so here we're trying a new innovative technique we called the Satinus lens technique. And we would take the sailboat, pick it up out of the water for this. This is having brought the sailboat back into the canal there. And on dry land, then we could apply this housing that we developed, and you can see the purple looking or pink looking lens, round lens there. And that's a type of plexiglass called Satinus. And when put up near to these optical sensors, it actually imitates the effect of water. And so by keeping it always, by being able to put it up to the sensors always the same location, same exact distance away from the face of the sensor, then over time we're able to tell if the sensors are always reporting, reasonably accurate over time, or were they drifting over time. And yeah, oh great, there we go. And what we found from graphing the validation data is that for the most part, it was actually really quite consistent. We took measurements, validation measurements before cleaning the sensors, so that there may still be any biofouling scums on there. And then we cleaned the sensors and we did an after validation as well. And so you can see in that image before and after, and for the most part, before and after were quite consistently near to each other in the measurement range. So that implies that we were cleaning them often enough. And it turns out that our 2-week cleaning schedule was about right. That seems to be a good time frame. So, okay, next slide. All right, so with the sensors ready to go and our operation set up, and we're there in Okeechobee, and we launched on December 31st, right into the new year, which was a really fun way to end one year and bring in the next. And you can see, hopefully that's showing there on your screen, you see Vela sailing off for the first time into the lake, headed towards the North Tower location. We ended up operating Vela for 287 days total throughout the year, and running multiple missions, each one lasting about approximately 2 weeks. And we launched from the very north end of the lake, where the Taylor Creek Lock is. So if you're not familiar with Lake Okeechobee, Lake Okeechobee has a large earthen berm dam that was built all the way around, the dike was built all the way around the entire lake to prevent flooding from hurricanes. And in order to get onto the lake, you go from a canal systems around the edges through a lock system and get access out onto the lake. So we were launching from the Taylor Creek Lock at the very north of the lake. At the end of each mission, we would charge the batteries, of course, give them a good charge, and do the Satinus validation checks, and then get her back out there as quick as we could, typically within about 24 to 48 hours. And we ended up collecting about 4 million measurements throughout the whole time on the lake. And then also what was really cool is that from the mission operations point of view, we were able to see what Vela was looking at in real time, in near real time. So let's move on to some of that. Next slide, please. Yeah, so here's the mission operations screen. And if you look at the right there, you can see the image, the L001 tower, we call the North Tower. And was the northernmost point of the triangular loop that we set up as our observing loop. And that primary loop, we came to call that the standard halo loop that Vela would travel. And the point of the loop is that it started at the North Tower where other instruments were, and then traveled around to other locations where the halo system, the halo overall program, was taking water samples and collecting other data, and also to other locations that were coincident with the Southern Florida Water Management District's observing systems that they have on the lake. And we wanted to repeat this standard halo loop as many times as reasonable throughout the year, so that we got a steady, mobile time series. So think of it as stationary time series, of course, is the Erlon system of sensors that were at the North Tower, collecting water quality samples in a stationary manner over time. But then introducing the idea of the mobile time series is a good way to think of it for the autonomous vehicle. And there on the left, you can see Vela starting out from Taylor Creek for the first time on December 31st, and the orange arrows indicates where the sailboat is and what direction it's pointing as it sails south towards the North Tower. I think there might be, Veronica, one little image there that if it pops up. Yeah, there we go. And she made it right out to the North Tower there. So next slide, please. Yeah, so from mission operations point of view, we were able to see the data coming in. And just for a moment, I want to make sure and thank the GCOOS, the Gulf Coast Ocean Observing System, for creating a public web portal where our data was being posted in near real time and for the public to view. And GCOOS is, of course, a partnering type of program with Sakura, all part of the Integrated Ocean Observing System. So yeah, so as the data was coming in, we could see hotspots of interesting data. For example, this is an O2 hotspot that we saw, and we weren't sure if it was just an anomalous data point or was there something bigger going on there. And so we sent the sailboat back to do a grid pattern there in that area, and you can see that grid pattern blown up there. And that in that case, we're outside cell range, so that's just satellite reporting back every 15 minutes, and so the grid pattern does look a little messy, but the point is we're able to survey that region right there and determine that it was an area of active rapid growth of algae. Okay, next slide, please. Oh yeah, we did the standard halo loop. We also did 3 side missions, so to speak. One was that we established a secondary small grid pattern that we did over time whenever we opportunistically could throughout the year, called the Taylor Creek Grid. We did a one-time mission to the Pokey Canal Point area and also a one-time mission to Clewiston. Next slide, please. Yeah, so this shows an image of the standard halo loop with the add-on of the Taylor Creek Grid at the very north end there. And we did that because in going to and from every couple of weeks, we crossed over that area and we found an unusual concentration of algae bloom up there, and we wanted to take a look at that more closely over the course of the year. So about once every 2 weeks or so, on the way in or the way out, we would stop and do that grid real quick and then head on to the rest of the standard halo loop. And we found some interesting things. Next slide, please. Yeah, here you can see that on the right side there, you can see the sailboat, and the bow of the sailboat is pushing through an algae bloom scum on the surface at Taylor Creek area, and that turned out to be from water sample. The dominant taxon was Dolichospermum, which is not Microcystis. We're most concerned about Microcystis aeruginosa, but this is another form of toxin-producing algae, sometimes called Anabaena because it produces a toxin called anatoxin. And it tends to form a very small colony size, a fine-grained colony size, but it appears, and from what we found here, is that it's very uniformly covering this area. And so if you look at the measurements, you can see a track there as the sailboat's turning a corner there. And the phycocyanin to chlorophyll ratio produces a very steady green look about it, just a basic look. And very little, not much variability at all, highly uniform. And that's pretty interesting. So from mission control, we got over time to get some experience in just being able to identify fairly quickly what the most likely types of algae blooms were. We started to get some idea of that over the course of the year. Next slide. Yeah, so on another side mission here, we did to Pahokee Canal Point area on the east side of the lake. And we had heard reports of a strong Microcystis bloom there, so we sent the sailboat there. And in the upper right corner, you can see green scum, typical of Microcystis. And also you can see that in the very bottom left down there. There's a nice little blow up of that too, that bright green color of Microcystis surface scum. And you can see in the background behind Vela there, the smoke cloud going up, which actually comes from the sugarcane fields there. There are sugarcane fields all around the southern area of the lake, and as a part of the way they operate, they grow for a couple of years, a few years, and then harvest, and then they burn the chatter, and then start over again. So at any given point in time, there tends to be a cloud in that area, and there's some concern about possible nutrient loading coming from the sugarcane fields getting back onto the lake and triggering Microcystis blooms. And it's just one thing that's being looked into. And interesting, if you look at the track line there, the blow up there of the sailboat sailing along there, you can see the phycocyanin to CHL ratio shows a lot of variability from strong purple to yellow, showing this considerable range of variability because the Microcystis turns out to be highly patchy in the way it grows. Next slide, please. Yeah, and here's our last side mission down to towards the Clewiston area. And here we really wanted to see if we could operate in the southern part of the lake because, as you can see in the little blow-up window image there are a lot of shallow areas, rocky reef areas, and dredge spoils there from the cross-Florida waterway goes through that area. And so we sent the sailboat from way far in the north straight down all the way to the southern area and got around those shallow areas and over towards Clewiston and back. So we did validate successfully that we could operate down there, which was cool. And we also scouted that area in advance first and picked up this photo of an active microcystis bloom. I thought I would include here just because sometimes these blooms look quite different. This is definitely a strong bloom, lots of well-developed colonies of an inch in size or more. And you can see them all mixed into the water there. So there was a little bit windy that day, and so the microcystis was not forming as surface scum, and so it looks quite visibly quite different. Okay, next slide, please. Yeah, so then we went back and continued doing the standard halo loop for the rest of the year and completed that as many times as we could to get a good track record of mobile time series. And finally, on January 1st, actually of 2022, Vela sailed back to Taylor Creek. And let's see, next slide. Yeah, so it went really well. We've operated throughout the whole year successfully. And the local community there of people, we got to know some folks quite well. We went out through the locks, as you can see in the upper image there on the left, a number of times, and there would be other folks going to or from the lake. And we got to hear a lot of stories about the lake, how it used to be clear back in the day, and how the algae blooms have come to become more prominent. We also got to talk to the lock operators on a regular basis, which was really interesting because they're there working right on the lake day in and day out. So they experience those blooms as well. And when they get intense, they have breathing problems and other issues. So it's actually quite something that people in the community are highly concerned about, and they know a lot about it. And they're very appreciative of the work being done, looking into to how to better manage and hopefully mitigate the algae blooms. I just want to say thanks to Dan Wood there in the bottom there. Well, that's my goofy look there, but that's Dan in the background there, and he helped out a ton. We did a lot of time down there working on sensor integrations and field missions as well. And thank you to the rest of the Navocean team. But more importantly, Marcia and Alan there just helped out immensely, and they helped us at times when we couldn't be there. And Alan took up taking care of the sailboat, launching and landing now and then. And Marcia, we learned so very much from you about all of the flora and fauna of the lake. And thank you to the rest of the folks in the Taylor Creek community. It was just wonderful being there, spending some time there, and thanks for all your support. So with that, I want to pass back to Veronica to talk about the smart stuff, the really important stuff, the science, and where that is going. So back to you, Veronica.
Okay, thank you. And so I just want to kind of like bring back the data in a look of how Vela measurements compare, or how they complement other kinds of measurements that we have of the lake. And so, as I showed before, like, yeah, we can get some information from remote sensing, for instance. And some of those products have like really large spatial coverage and very high resolution in that. But the issue is that we don't always get such clear images. And most of the time in the summer time, when we have bloom problems, we get a lot of clouds and glint. And so when we get the time series of that, there's a lot of information that is missed. So we really don't know the temporal aspects of it. So we don't know when and where a bloom starts. We don't know if it moves and how fast it moves. And we don't know how long it lasts. And I will be showing you some like spatial reconstructions of stuff from Vela later, and so some of these questions can be answered later. And so from the monitoring, traditional monitoring stuff, as I showed before, yeah, there are plenty of stations in Lake Okeechobee. There are plenty of effort on that, and some of those stations have a pretty long temporal coverage. But again, even if we have two close stations monitoring, we lose spatial structure, especially for instance, the microcystis, as Scott showed you, those really patchiness of those signals. So we don't see those small patterns. And when we get kind of like statistical results of this, yes, we can get like a number of occurrences of blooms, and this again taken from the last year report, 2020 report. Number of occurrences of blooms, so like in stations and in different coastal stations, in out of water stations, but we don't really know the small temporal things. For the continuous monitoring stations, here I am showing 2 of the newly installed aquatrolls from the DEP, and there's one here at the L001, and there's one down here in LZ40. So we can see them moving around, and we can see how they change. But you can truly see that they have different patterns. They're totally different and then behaving differently. So what happens in between those things? We don't really know, and we cannot account for that variability. And this is something that I think a lot about. And so here is a kind of a comparison, kind of like to bring your mind to that, and how we can think about that, and how we can think about what data points or data sets represent. And so here again, L001, we have this red dot, and on the most right panel, we have, so all these are time series, and so this is a traditional monitoring time series bi-weekly. So people go to the field. It is the best kind of accuracy because this is actually samples. So they go to the field and they take samples. This is like the time series that we get. Next to it is directly the continuous monitoring system from Oracle, or for the system that we deployed, and you can see the same spot, the same thing, and you can see all this variability, all these things going on. And so you can clearly see that if you were there on the field one day before or one day after, or 1 hour before, 1 hour after, you will get a totally different picture of what's going on. And it's quite different. We miss all those things. And here on the next, on the leftmost slide, you have a depiction of the transects extracted from Vela from L001 here, that this corresponds to this side of the graph, the right side of the graph, to down in the kind of like the center of the lake on the right side, on the left side of the graph. And so all of these lines correspond to those transects. And so this is, again, all this chlorophyll, and you can see like high chlorophyll in yellow developing here close to the L001 station, but you also see blooms forming in other places, like for instance in this region, which will correspond to somewhere here in the middle of that transect that you are not seeing for these monitoring strategies. So to me, it's always very important to think about variability and what it is, and how do we interpret the measurements that we take. So, and this counts for harmful algal bloom, but also for all the variables related to harmful algal blooms. So, because at the end of the day, what we want is to quantify biomass, and we want to quantify biomass lake-wise. So recently we were on a conference that they were talking about how to compare lakes. And so when you quantify the biomass of an entire lake, how do you do that? Based on what values? And so one approach of doing that, and that was also part of the HALO project, is to implement models. So we can implement a lake-wide model, and we can try to validate that model. But to get a good model, we need initial conditions, we need fluxes, we need parametrizations. And if we base our parametrizations based on snapshot measurements, what are we really doing? So I think a lot about how do we capture small-scale patterns and variability, and how do we relate that. So the ASV allows for a very large area coverage and very high temporal resolution, but the challenge in here is that it is a space-time composite. So how do you detrend that, or how you take it out? And so this is a way to look at it. So here I am presenting different time series. So on the top, it is the Vela, the sailboat time series for chlorophyll. So all these 3 time series for chlorophyll, starting second half of June and ending in July. This is kind of like a 1 month measurement. And so you see here for Vela, it is only, I have only kept the data that was collected in a 1 circle radius from the L001 tower. So those data is kind of comparable. So every one of these spikes in here that you see in here, and these spikes are actually a lot of data. They're comparably really close because we're talking minutes here. And when we compare that to the continuous monitoring station, this is our station, the HALO station at L001, and this is the DEP station at the same spot. So we kind of see the same pattern, right? So you see this pattern in here, and you see like this kind of like elevated pattern in late June and early July, and then it drops, and then it starts growing up again, and then it starts growing up again. kind of see the same pattern, but there's a lot of things in space that we are missing. Even if we look just at that, like just at this point, so this point represents all the variability that was captured within a 1 kilometer distance from the sampling from that L001 tower. So when we look at the entire data set, what do we see? And so I want to point out that what we don't see in the temporal realm, by conclusion, should be spatial, right? And so one of the things that we see clearly in this is for Vela, this in this green mesh color, what we have is this is the full data set, like all the data transmissions after cleanup. And then we have a kind of a 15 minutes running mean and a 1 hour running mean and 1 day and 3 days, so we can kind of like compare. And so if we compare, say, the 1 hour time space series with the 1 hour record that we have from Lobo or from the DAP, we see these spikes. We see kind of like this recurrent every 2-3 days spikes, things that occur, and we don't see those things happening in the temporal series. So this is basically related to the spatial variability and the sizes of the patches and the things that the vehicle crosses in time, and they are pretty consistent, and we see them appearing. So even when comparing, and again, 2 different stations, so we have the DAP L001 and L040, and I showed you before that they were quite different. But even if you monitor and compare those 2, you are not actually capturing this variability that we see in here. So it is really important to think about that and what it represents and how do we look at it. So this is a kind of a close-up of our data and what we saw like during a month of monitoring. What did we see during the entire year? So this is the record of all the full data sets of more than 4 million data points for a set of the sensors. So here I am showing you the full data set and the daily mean for that. On the top, we have the backscatter that you can read as turbidity, chlorophyll A in here, then followed by phycocyanin, CDOM, which is the dissolved organic matter, the colored portion of the dissolved organic matter, conductivity, temperature, and oxygen. And some things from these patterns that we can see. First, we see like clear seasonal patterns where we expect them to see. We see the temperature increase and decrease as expected is higher in summer. We see the conductivity changes, but what about, and we see the oxygen in general, right? So like, there's a dip in oxygen over the summer. But what about the other things? So there's really super high variability. There's like extreme peaks in here and things that go up wildly, and there can be changes in the thousands for that of what we are representing. We still, if we consider just like the daily means, we still can see some patterns. We can see these peaks that I was talking about, and this is related to the spatial structure of that. But some of the things that I want to point out, we detected blooms in the chlorophyll and even in the phycocyanin pretty early, very, very early, before like they were detected by any other monitoring attempt. So since February, even like as early as February, and sometimes a little bit as early as in January, were really strong signals of phycocyanin in June and July in here, then to decrease a little bit, and then to have like a second kind of spike period later in the year. But what I want to point out is that this high frequency variability is way higher than the seasonal trends. So these things that we see in the fluorometer measurements are much higher than the seasonal trends that we have. And so we must think about what it does mean from the space perspective. So kind of like switching to the space dimension, how do we think about the space dimension? And here, I guess that's the only time, the little thing that I'm going to talk to you about, like what our data shows. There's much more, but we don't have the time. So how can we use these to seek spatial structure and spatial movement of things? So for that, we just chose the regular loop of that, and I constructed some geofences to constrain the data. And so here you see 3 different geofences, which I call like the left transect or the coastal transect, the bottom transect over here in white, and the right transit in magenta. And so if we construct these geofences and then we trace a line in the middle of it and then we project the data into that line, we can think about that as just space. And if we repeat that line in time, we have a space-time dimension. So here's an example of what I'm talking about. So this is the coastal transect, or the left transect in here. And so this is time series, the entire time series for the entire year, starting in January, well, in December 31st here and finishing in January 01 and 2022 in here. So this is time. These are the different variables: chlorophyll, phycocyanin, CDOM, and backscatter. And here we have the transect over it. So the bottom of the transect, it will be the bottom of the transect over here, and the upper part of the transect will be the upper part of the transect here. And so what can we see here? Some really exciting things. And so first of all, you can really see this bloom starting to appear here in the chlorophyll pretty early. Not that much of phycocyanin, but very high chlorophyll. And so we have those blips and we went to see. And again, as Scott said before, those earlier blooms were not exactly microcystis, but there were others, and some water samples at that time confirmed that this was Dolichospermum and Cylindrotheca mostly. Some other blooms appeared like later in the year that were like mixed flagellates sometimes and other species and things. And then a high phycocyanin signal. And something that I want to point here. Here we see this signal, pretty high values, but they are detached. So these are kind of like two different populations. It's not the same thing. Pretty high and short lasting, and they are not connected. But what is interesting about here is to see these slopes. What does a slope mean? So this slope is motion. Why is it motion? Because as this is the time dimension and this is the space dimension, and space or time that goes velocity. So this is basically motion. So this motion, it says that there is a bloom originating down here and moving up here at a velocity of about a few days per kilometer. And so that motion in the map, what it looks like is this. So we registered the bloom forming down here and then slowly, well, not slowly, pretty fastly moving up here. Then we have some blooms appear very sporadically here, like fast and very high concentrations, but they die off. And then again, another slope. We have the algae forming down here and then slowly moving upwards. And then we have a blob. This blob is not motion, this blob is growth. So as it is not, we see as it expands in every direction. So this is a blob forming here and then growing down. And the rest of the year, pretty high concentrations, but then we see another motion, another slope, but this is in the other direction. This in the map corresponds to a direction in this, where the arrow is pointing, so it's the reverse of what is happening, and it again happens later in the year. So there are many things to comment about, like how, for instance, how variables correlate. So we sometimes have high phycocyanin signal and relatively low chlorophyll, or sometimes relatively high chlorophyll and relatively low phycocyanin, and these can be used to describe populations and things. But there's not much time to talk about that. There are also some signals in the CDOM that they occur kind of at the same time to this, but there's actually a temporal lag in between them and things are happening. But let's move to another transect. So the right transect, this kind of like more centered transect than what we can see here. Again, we can see blooms forming in the phycocyanin here, and they are kind of like detached populations. They are not joined, and so we see pretty high, pretty patchy, as Scott showed before. So it's not just yellow, but you have like yellow, blue, yellow, blue, yellow, blue. So those are like pretty patchy and pretty big blooms. And then you have a slope, but this slope is less steeper than the other one. This means a much slower motion than in the previous case, and this motion in the map corresponds to the direction this arrow is pointing. It's from up here north, kind of like to the more inner lake in that. And these things happen. What about the bottom transect and what's happening in there? Some other interesting things that happen. So here you have this slope. There's an earlier slope in here, but this direction, for instance, of motion that we can detect here corresponds to this motion in the map where the arrow is pointing. So the bloom is originating here, And I mean originating in a very broad way, because this bloom actually can be moved here from other places. But it appears here in the middle of the transect, and then it moves towards the canal in here. So this is the really first time that we can see bloom motion in real life, in real time, how it happens. And I think it's pretty interesting to explore it, and how the dynamics in terms of days, in terms of hours, in terms of patchiness, how they move about that. So I will kind of like stop about here, because it could ramble about that for hours, but we don't have that much time. So I wanna just like let some like highlights about that. What Scott talked about. Yeah, we were pretty successful with Vela. It operated successfully. We integrated successfully a suite of packages, and Jordan wanted to make sure that I said that there are not that many sensor packages outside in commercially available that can monitor HABs directly. And so using packages, using commonly used sensors in a kind of like clever and integrated way, and how these variables behave, interrelate between others, is one way of approaching these. Another thing that I want to point is that the Satinus validation that Scott talked, it is a good good way of validating things. It's a consistent method, and so other people using fluorometer validation in field can use that. We could identify blooms. We could identify complex relationship. We could identify that variability in space is much larger than variability in time. And just this last slide, I just wanted to include like an acknowledgement slide. This is one effort of a lot of people. First of all, the funding. Without the funding, we won't be able to do that. And so we got that from the DAP. And all the people that made it possible, from Harbor Branch, all the people that worked in the project planning, in the field validation, and the lab work, and in the data processing, the Navocean people, the Navocean folks in the boat design construction, and also like GCUs that worked for the deployment for the portal of the operations. And I guess I will leave it in here. Thank you.
Great, thanks so much, Veronica and Scott. And as a reminder, use the tablet looking icon to ask questions. We've had quite a few come in during the presentation, so I'm just gonna dive right in. This is from Matthew. Were any elevated findings reported to South Florida Water Management District or the Florida Department of Environmental Protection as part of their algae response efforts? Jordan, if you're talking, you are on mute.
Hey, yeah. So yeah, hi all. I pawned this presentation off on Veronica because I figured it would be great exposure for her, and she has really led the the integration of a lot of this data. So it just seemed more appropriate that she was the presenter. Did we inform DEP? You know, it was we definitely did, but it was more anecdotal, right? So it wasn't, we didn't really establish, let's say, a formal mechanism in doing this. Yeah, so I mean, there was probably a half dozen instances where we let DEP know that there was indeed some type of bloom activity, especially early in the season, right? So Scott being out there on the lake so frequently, it really helps having somebody out there. He was out, probably in the water realistically once a week, checking on the boat or whatnot. So he got to see early on these blooms that popped up, as early as February, which was pretty surprising. So we did notify DEP, but from the idea of let's say informing mitigation action or public health, probably not so much.
Thanks, Jordan. We have a question from Dave Jones. How many sailboats do you think are needed for adequate monitoring?
Good question. Jordan, do you want to get that one again?
Oh, well, we looked at just the total distances, and this is actually something also that Veronica, a lot of her work on the spatial dimension, we have some ideas for how to treat the data. almost like if you could reproduce a grid. Let's say you could make an imaginary grid that defines the spatial variabilities that we observed with the sailboat, just in the data that we collected, right? If you could define some imaginary grid and then let's say also define some acceptable level of acceptable variability that you would want in order to try to conduct some mitigation action for HABs, or to ensure that you're conducting sufficient monitoring. How many either stationary sensors or how many sailboats would you need? And that's something that she's working on as we speak. But just from the sake of having lake coverage, I would venture to say that we had pretty good coverage just from having one sailboat in the north of the lake. So I think you could get away with having two. I mean, it was from the last slides that Veronica presented, it was pretty clear that even just having this triangular transect, this triangular pattern, and you would hit each of the triangle transect legs, on the order of every few days, you could still make out the motions in the pattern. So it kind of depends on your acceptable level of, informing early action. But I think, having two would be pretty ideal just to get full lake coverage.
Great, thanks so much.
That's a great question.
And this question is from Paul Gray. How many years do you plan to use the boat? Is there continued funding for this project?
As of right now, there is not actually. So that's something that we're still working on. We're also trying to leverage we're trying to leverage new funding sources. So yeah, we just received this news the other day. So yeah, we're still trying to pursue it, but as of the 1 year was just what we were funded for, and I think it was one of the most successful aspects of the project. I think the data speaks to that.
And another question from Dave: do you see the sailboats being a potential early warning platform to trigger other concentrated monitoring or deployment of additional platforms?
Veronica, you want to go? Scott, you want to go?
Sure. I think I could speak to that a little bit. That's definitely one of the uses that we have in mind for our sailboats and for the data collected there. I think if you look at use as an opportunity to kind of expand the vision of the concept a little bit, that's for Lake Okeechobee. We can certainly use them in that manner to figure out where the blooms are most intense and then to track the development of those. But it's also relevant to the coastal oceans as well, and estuaries, where there's a lot of work going on now from many different institutions who are working on models to try and, from a modeling objective, be able to anticipate and predict the coastal ocean environment and the HABs pollution there. And so the kinds of motion detection things that Veronica is working on potentially be very useful to help with an early warning type of system. Yeah.
Great, thanks, Scott. And another question from Dave. It looks like the data can be made available pretty quickly. How long does the QA/QC take before we could perhaps share it via GeoCollaborate?
Veronica, sounds like a good one for you.
Well, I'm not sure how can I answer that. So the QC of the data is pretty much done at this point. I think mostly there are some concerns about, like, for instance, filtering the data. What Scott was saying, that the data pings every 5 seconds, and then it stops, and for 25 seconds and so on. So there are some things that you have to do to clean up, and when you get like zeros for no data and things like that, that's pretty easy to do. And I think, like, yeah, the most challenging thing is to trying to recognize if the peaks that you see are real or not, because of the turbidity of the lake. And I think from what we have seen in the field, they are real, they are in there. So we trust that totally. And so we currently are working on a methods paper to get out the results as soon as possible that we're going to be publishing. And once that is out, I don't know, I think it's up to Jordan on when the data becomes available to the public.
Well, it's
Great.
already available, right? So it's already available via GCUS.
Oh, well, yeah, the transmitted data that is kind of like every 15 minutes, it is.
Yeah, that's right. Yeah. So the QC data is coming. Mm-hmm.
Perfect. And we did have a lot of questions about how people can access the data or look at it in real time. So maybe we could share that GCUS link to everyone that has registered. And seeing that it's 1 o'clock, we actually have quite a few questions that are unanswered. So maybe we answer one last question, and then everything we didn't answer, I send you guys a follow-up email, and we'll get the responses to those who asked. Does that sound like a good plan? Perfect. This question is actually for Scott. Somebody had a question on the cost of the sailboats, and then also, do you think the sailboats could be effectively applied in smaller water bodies? Is there a minimum wind speed required?
Okay, good. Yeah, so cost-wise, depending upon the sensor suites, we're moving from development to application to selling boats, and so they're in the range of around $100,000 to as much as $200,000, depending upon the sensors integrated. The sensors tend to make a big difference. And as far as how small of a body of water could they be used on, and well, that really depends on the science objective. So basically, the sailboats can also be used. That's something that I think I forgot to mention in the talk, was that they have also an electric thruster on board too. So the electric thruster is used for backup propulsion in the case of no winds, or in the case of just needing some speed. And also that provides some, if you want to just do a short range deployment in a smaller body of water, that could be useful as well. But the depth right now in the current design requires about 31 inches of water depth, but in the next iteration of the design, we'll be making a modular keel that can be reduced to a shallower depth. So that's really the primary, how deep is a small body of water? That's really the primary limiting factor. But the next design will be able to accommodate about 1 foot depth or less.
Scott,
I think there's a question with just the minimum wind speed. Before I forget that, in wind speeds above 3 nautical miles per hour, we start to get slow forward motion that's beneficial, and about a half a knot of speed forward. So anything from 3 up, you start to get useful benefit from the wind.
You showed that picture of the storm in the Banana River Lagoon.
Yeah.
On in your presentation. So the Banana River Lagoon is what, less than 1 kilometer wide and maybe 3 kilometers north to south. And with a 48 knot wind or so, the boat was successfully able to navigate within that small basin over the course of 3 days or so. So that's a pretty good example, I think.
That's pretty incredible.
Yeah, that was pretty exciting.
Yes.
And we were happy to see how well it worked. It was outdid our expectations, so that was fantastic.
A great test of the technology. Well, thank you all so much for joining us, and thank you everyone for attending and taking their lunch hour with us. We didn't get to a lot of questions, but you'll see a follow-up email, and in a few days we'll also send the recording. And if you have any questions, please reach out and we'll be sure to pose them to the presenter. All right, thank you everybody. Enjoy your Tuesday. Bye-bye.
Thank you
so much, Abby.
Thanks, Abby. And take care.